Welcome to BRC Analytics, a portal for genomic data on pathogens, hosts, and vectors. Ultimate goal here is to include all species for which genomic data is available. And you will be able to explore these data sets here and also perform analysis, no matter how complex or large, and always for free. So for example, if you have RNA-seq data sets that you would like to analyze, you will be able to do it here. If you have resequencing data where you want to identify variants, you will be able to do it here. If you have surveillance data for a particular pathogen, you will also be able to analyze it here. ATAC-seq, ChIP-seq, genome assembly, anything you can possibly imagine. BRC Analytics is built around two fundamental concepts: organisms and assemblies. So organisms are simply all the species that are included in this resource. And at this time, you can see we have about 1,100 of them. If you click on any species, or if you search for a particular species, let us, for example, search for Plasmodium vivax. So here is this single species. If you click on that species, you will see all assemblies associated with that. And assemblies are individual genome builds. So for example, in this case, these are builds for different strains, for example, for different geographic isolates. In other words, an assembly, it's a particular genome, a particular version of a genome for this species. And this page provides some information about how good these assemblies are. For example, it tells you whether it's a reference assembly according to NCBI, and it tells you at what level this assembly is. So the best level is, of course, the chromosome level. The next best is scaffold, and well, the worst in this concept is contig. So these two assemblies are chromosome level assemblies. They are good. This is a scaffold assembly, and you have two contig level assemblies. And this is their length in nucleotides. Each assembly has two controls associated with it: the Analyze button and View button. If you click View button, you will be able to, for example, view annotations for this particular assembly at UCSC Genome Browser. And this would include information about genes and other genome features for this species. If you click the Analyze button, you will be given a list of analysis choices that you can perform here. The next series of videos will describe each of these analyses individually. Before we finish, I would like to emphasize that if you want to add organisms to this list, or you would like to add gene annotation or annotation of regulatory regions, of anything that you would like to add, you should use this help functionality, which will point you to this feedback page where you will be able to tell us, please add this.